in the cutting-edge research labs of Genomic Solutions, a company at the forefront of biotechnological innovation, the atmosphere buzzed with excitement and the hum of advanced machinery. The company was known for pushing the boundaries of genetic engineering and personalized medicine, making strides that could change the face of healthcare. In a sleek, glass-walled conference room overlooking the bustling lab floor, Dr. Ava Thompson, a senior geneticist, sat with Dr. Michael Carter, a young bioinformatics expert. They were deep in discussion about their latest project, which aimed to develop a groundbreaking gene therapy for a rare genetic disorder.
Michael, as we dive into this new project, I've been reflecting on the challenges we're likely to face. Developing a gene therapy for such a rare condition is no small feat. What do you see as the biggest obstacle we need to overcome to make this a success?
I've been thinking about that too, Ava. The complexity of the human genome means there are countless variables at play. But from your perspective, what's the most critical factor in ensuring we can deliver a safe and effective treatment?
The most critical factor, in my opinion, is precision. We need to ensure that the gene editing is as accurate as possible to avoid off-target effects, which could lead to unintended consequences. Question: Michael, what do you think is the most crucial element in developing a safe and effective gene therapy? A. Achieving precise gene editing to minimize off-target effects. B. Ensuring robust clinical trials to thoroughly test the therapy's safety. C. Developing efficient delivery methods to target the affected cells accurately.
Answer: A. Achieving precise gene editing to minimize off-target effects.
Michael, I believe that precision in gene editing, option A, is paramount. If we can't guarantee that we're only targeting the intended gene, we risk causing more harm than good. Precision ensures that we're delivering a treatment that's not only effective but also safe.
That's absolutely true, Ava. The margin for error in gene therapy is incredibly small. We need to use the most advanced tools available to ensure our edits are exactly where they need to be.
Exactly. And by focusing on precision, we're setting a strong foundation for everything else in the project.
They continued discussing the tools and technologies they would employ to ensure the highest level of precision in their gene editing process. Ava emphasized the importance of using CRISPR-Cas9 and other advanced techniques to achieve their goals, while also considering the potential for future advancements in the field.
Ava, while precision is key, I am also concerned about how we're going to deliver this therapy to the right cells without triggering an immune response. What's your take on the best strategy for that? Question: Ava, what do you think is the best strategy to ensure that the gene therapy is delivered effectively and safely? A. Using viral vectors to deliver the gene therapy with high specificity. B. Developing nanoparticle-based delivery systems to avoid immune detection. C. Exploring non-viral delivery methods to reduce potential side effects.
Answer. B. Developing nanoparticle-based delivery systems to avoid immune detection.
Ava, I think that developing nanoparticle-based delivery systems, option B, is a promising approach. Nanoparticles can be engineered to evade the immune system and deliver the gene therapy directly to the target cells, reducing the risk of side effects.
That's a great point, Michael. Nanoparticles offer a level of precision and control that viral vectors sometimes lack, especially when it comes to avoiding immune responses.
Exactly. And if we can perfect this delivery method, it could open up new possibilities for other therapies as well.
They explored the latest advancements in nanoparticle technology, discussing how these tiny carriers could be customized to deliver genetic material to specific cells without being recognized by the immune system. Ava pointed out that this approach not only had potential for their current project but could also revolutionize the delivery of other treatments in the future.
Michael, another aspect we can't overlook is the ethical implications of what we're doing. Gene therapy, especially for rare conditions, raises a lot of ethical questions. How do you think we should address these concerns as we move forward? Question. Michael, what's the most important ethical consideration we should keep in mind during this project? A. Ensuring informed consent from patients and their families. B. Balancing the potential benefits with the risks of unforeseen consequences. C. Considering the long-term impacts of gene therapy on future generations.
Answer. C. Considering the long-term impacts of gene therapy on future generations.
I believe that considering the long-term impacts, option C, is crucial, Michael. Gene therapy doesn't just affect the current patient. It could have implications for future generations. We need to think carefully about the potential ripple effects of our work.
You're right, Ava. The changes we make could be passed down to future generations, so we need to proceed with caution and a strong ethical framework.
Exactly. By addressing these concerns head-on, we ensure that our work not only advances science but also respects the potential implications for society as a whole.
They discussed the ethical frameworks that would guide their research, including the importance of transparency, patient education, and rigorous ethical review processes. Ava emphasized that by considering the long-term impacts of their work, they would be able to make more informed and responsible decisions throughout the project.
After discussing all of this, Ava, I'm curious. What's the most important mindset we should foster in our team to keep this project moving forward successfully?
The most important mindset is one of innovation and caution. We need to be bold in our ideas and approaches, but also meticulous in our execution to ensure that we're advancing the field safely.
That's a great balance. Innovation drives progress, but caution ensures we do no harm in the process.
As their meeting came to a close, both Ava and Michael felt a renewed sense of purpose. By focusing on precision, innovative delivery methods, ethical considerations, and a balanced mindset. They were confident that their project would not only succeed but also set new standards in the field of biotechnology. With their strategy clear and their goals aligned, Ava and Michael left the conference room ready to push the boundaries of what was possible in gene therapy, knowing that their work could one day change the lives of countless patients around the world. Here are 5 key concepts related to the theme of biotech innovation.
Precision. Ensuring accuracy in gene editing to avoid unintended effects and deliver safe, effective therapies. Innovative delivery methods. Exploring nanoparticle-based systems to improve the targeting and safety of gene therapy.
Ethical considerations. Weighing the long-term impacts of gene therapy on future generations and society. Informed consent. Prioritizing transparency and patient education to ensure ethical practices in experimental treatments.
Innovation and caution. Balancing bold ideas with meticulous execution to advance the field responsibly.